Okay, thanks for having me, and thanks to the organizers for the great event. It's been a blast. So today I will talk about the population genetic dormancy, but the title should have been more on the follow dynamics of dormancy. So, wait, it's not moving. Okay. So dormancy is a popular bed hedging strategy of a life cycle trait employed by a lot of organisms. So the popular example would be the plants, where they under environmental fluctuation, they go into the seed, and then when the spring comes up, then they germinate and so on. And then the other examples are microbes, where the fungi or bacteria, they stochastically transition back and forth between the active state and dormant state. And I guess like one of the main motivation for us was that when we study the Mycobacterium tuberculosis, it has very long latent infection period, so that the infectious disease tracking as well as the studying evolution of this tuberculosis has been very challenging due to this long latency period. And even under neutrality, it has really a huge impact or important impact on genetic diversity of the population. So for example, depending on how long it stays under dormancy, it affects on the time to reach mutation drift equilibrium and thereby the genetic diversity. And you can see under this expected site frequency spectrum, the black is the under dormancy model, and then the second dark is the two-island structural coalescence model, and then the lightest one is the Kingman coalescence model. So you can see the dormancy can actually have huge impact even on the genetic diversity and its expected site frequency spectrum. So there has been two major coalescence models employing or accommodating dormancy. So one is something called weak seed bank coalescence, weak in a sense that the time spent in dormancy is negligible with respect to the coalescence time scale. So this model can be utilized for the annual or perennial plants. So the model, instead of picking your ancestor right before the previous generation, there is some geometric time distribution over which you can select your previous ancestor. So statistically, this has been shown to be equivalent to the Kingman coalescence, but with a delayed coalescence time. So in terms of inference problem, one can utilize the inference method developed for Kingman, but there's some identifiability issue about whether the dormancy or the slowed mutation rate. And this is like one of the main focus of this current talk, which is something called strong coalescence model. So strong in the sense that the time spent in dormancy is in the order of population size. So one of the major characteristic here is that it has two compartments, active population and dormant population. And lineages can switch back and forth between active and dormant stochastically. But then in the dormant population, you cannot reproduce. That also means that in the coalescent model, you cannot coalesce if you're in the dormant population compartment. So this is intrinsically different from structural coalescent model. So in two population structure coalescent, whether you're in population 1 or population 2, you can coalesce as long as you're in the same population. But for a strong coalescent model, seed-bank coalescent model, there's an asymmetry in coalescent process where the inactive population you can coalesce, but not in dormant. So even in terms of forward Wright-Fisher diffusion to population structure coalescence, there is a migration term in both, and then the drift term in both population 1 and population 2. But then the strong seed-bank coalescence, there's a type change active to dormant, dormant to active in both populations. But in dormant population, there's no drift term because there's no reproduction, and also thereby there's no coalescence. So more formally, the strong seed-bank coalescence, from now on, I will just say strong seed-bank coalescence, can be stated in terms of the marked partition on number of samples. So marked mean that the partitions are labeled whether it's active or dormant. So in this example, we have everyone in the initial condition, everyone is in singleton, and in this example, everyone is in active label, but it doesn't have to be. It can be either active or dormant sample if you can sample from dormant population. And then because the coalescent event has to be between in the active individuals, so the observing state at the root, everything is in one block, but the label has to be active label. And then in between, there are three possible processes. So transitions. So one is the merging two active blocks. So again, timing goes from backward in time. So tip to root. So the coalescence between active blocks happens with rate 1. And then the type switching from active to dormant is happening with rate C. And then type switching between dormant to active happening with the rate C times K. This parameterization is so that we maintain the constant population size. So this is a realization of the strong seed bank coalescence process. So in seed bank genealogy, it's a color genealogy. The jump chain defines the color topology, and then the waiting time between these transitions define the branch length. And because we are also considering infectious disease application, so we also extend this model to have the heterochronous sampling. So to build inference framework, I'm a Bayesian person, so we are doing Bayesian value dynamics inference. So as Professor Aaron King excellently explained, the generative model in value dynamics, so it's different from phylogenetics, where in our case, value dynamics, the ultimate goal of the inference is the underlying population dynamics parameter from which we have genealogical process where we generate genealogy, which relates to who is related to who ancestrally. And then given the genealogy, we superimpose under neutrality assumption, we superimpose the mutational processes, and that generates our observed data that we want to infer from. And then the inference is the inverse problem. Given our data, we want to target our target posterior is that the genealogy and that population dynamic parameter and the mutation model parameters. And if you're not interested in genealogy, you can marginalize them out by integrating over. So the following inference with the seed bank coalescent, our target posterior is that we want to infer jointly our seed bank genealogy. And then our seed bank model parameters, K relative population size active to dormant, C transition rate from active to dormant, and then the scaled effective population size of active population. And then we have substitution model parameters. So we have two substitution model parameters that I will explain later why. So we have one for active population and one for dormant population. And this is our data. And then usually you can incorporate other metadata associated with your genetic data as well. And then each space rule we can decompose into these three terms. First is a genealogical prior, where we're going to use a seed bank coalescent model, or if you're birth death camp, then you can use a birth death sampling process as well. And the second term is the phylogenetic likelihood. So this is if you are familiar with the like IQ tree, maximum likelihood estimation of phylogenetic tree, this is what people are maximizing to get the phylogenetic tree. And then third ones are just other priors that are relevant for Bayesian inference. So, the seed bank prior under seed bank coalescent model, we derive that this is the genealogical prior under seed bank coalescent model. So this looks fanciful, but if you actually just tease apart, it is very simple intuitive explanation. So if we partition our time into the time intervals, where for each time interval, each end is one of these 4 events, and then in between the interval, in between event time, nothing happens. So this is just the probability of nothing happens in between the events, and each color terms contribution are colored by the event type. And then at the end of time interval, there's something happens, one of these events. And then sampling is deterministic because we know sampling time, so we have the probability 1. So that is our genealogical prior, and then the phylogenetic likelihood term. So this was actually interesting part for us because if you're familiar with the standard structure coalescent model, usually in computing likelihood, the color is actually ignored and the likelihood is computed as if they're not colored, because assumption there is that the population location doesn't affect on the mutation rate. However, dormant population in our case are usually often mutating slower than the active population. So we have to consider coloring and model them into different mutation rates when computing likelihood as well. So we kind of, maybe this is not worthy, but we proved that the likelihood under seed bank coalescent depends only on the substitution model parameter of active, and then an edge length, and as well as the total dormant state proportions per each branch. So intuitively, this means that for each branch contribution to the likelihood, it doesn't matter whether this total dormancy length is in one contiguous chunk or divided into the multiple small chunks. As long as their total duration is the same, the likelihood contribution is the same. So for each branch. So that means that for each branch, we can define the dormancy length weighted effective mutation rate for each branch, and then we can utilize it to compute the likelihood. So if you're familiar with the branch-specific rate model or the random clock model, so it has some connection to there as well. So it can be very efficiently computed with the standard Falsenstein pruning algorithm, which is dynamic programming, because we prove the likelihood can be computed with effective mutation rate per each branch. So we don't have to visit all these type change nodes. So we can only traverse only the internal coalescent node in computing this likelihood. So the inference is done under the standard reversible jump MCMC. So here the Metropolis Hastings Green's factor comes in. When we propose the new tree and then they have different number of type change events, then they actually change underlying tree dimensionality, space of the dimensionality. So that's where the Green's factor is there. So the other parameter, like the scalar parameters, I mean the real number parameters, is easy to explore because like the standard transition kernels can efficiently explore the space. However, the tree space is very, as you may all know, it's very large. So there, and now we have color tree space. So we need to be able to explore the color tree space in a very efficient way. So for example, this is like our sample run. Coloring scheme is slightly different. So you have to be able to propose the tree from the given current tree and then compute the acceptance probability and then accept or reject and move and so on until the stationary distribution is achieved. Oops. So the question is, so given the current color tree, like how do we propose what's the next color tree to explore efficient way, meaning that they try to minimize the mixing time. So we tried many different approaches, but like we found this empirically, this approach worked the best. So first we uncolor the tree. So we strip out all the coloring of this tree. And then we apply the standard uncolored tree operator. For example, in this example, subtree prune regraphed. So this subtree is pruned and then reattached to this site. Oh, sorry. Sorry. I sure doesn't have any one slide. They're all anyways. And so that don't scare you. Okay. Okay, so once we have the uncolored tree move, then we need to color the affected edges. So we can actually transfer for the unaffected edges. We can just recolor the unaffected edges. But for example, for this particular branch edges, the actual branch length and then the topology has changed. So if we were to just vanilla apply the existing coloring, like port over to the new topology or new uncolored tree, then this is not valid. First, this is a coalescent node, so it cannot be active. And also the branch length has been changed. So the number of event and timing of this type change event should be resampled. So we now have to recolor them. So how we recolor it? So we sample the path from the endpoint conditioned continuous Markov chain. So for example, but if our chain sample from the chain is like this, it's valid. But if it's like this, because like both ends have to be active for the internal edges, then this is not a valid sample. So what we do is that we use I don't know why it's still there. We use something called uniformization technique, which is a very popular technique in sampling the endpoint condition continuous time Markov chain. So we first define this something called uniformization rate, which is the dominant exit rate from your rate matrix of the continuous time chain. And then we construct a discrete time Markov chain using this uniformization rate. So the basic idea of this uniformization is that you convert the continuous time Markov bridge problem into two sub-problems, conditionally independent. So first is a Poisson grid, so sample n number of event number, and also the event time of this conditional on this event number. And then once we have that, then we need to sample the event type for each of these events. So we use this using the discrete time Markov chain, Markov bridge. So slightly more detail in sampling the number of events. So this is the Poisson distribution, standard Poisson distribution with the uniformization rate and length. And then this is just tilting or weighting of our probability based on the endpoint conditioning of our discrete chain. And then once we have the number of events being sampled, let's say 3, given conditioning on the number of events, the homogeneous Poisson process, the event time is uniform. So we just sample uniform event time from the IID, uniform from 0 to L. And then now the only thing we have left is the assigning the event type for each event. So we use the standard forward filtering and backward sampling using the conditional transition kernel from standard HMM discrete time Markov chain literature. So this is example, and we are going to have the self loop. That's why I call virtual event, because if you look at the construction of the discrete time rate, there's going to be a guaranteed to be at least one of diagonal element being 0. Okay, so last one, because we have the discrete chain path to construct, going back to the continuous time path, we remove the self loop, which is a virtual event, and then we merge those interjump holding time to get the discrete continuous time sampled path. So in summary, step 1 of this tree proposal in the color tree space is the on-color tree move, and then the recolor affected edges using our uniformization scheme. So we did extensive validation using the synthetic data with the known ground truth parameters and tree, but then it may not be as interesting. So like application, we apply to tuberculosis data. So tuberculosis, once you're infected, you can have very long, up to 10 years of latent infection because at lung, it's encased by these immune cells, so it can be inactive for a very prolonged period. But it is very hard to find a lot of literature or experimental data studying this latent infection mutation rate and latent infection process in vivo. So this is one of the few that we can find. So it is studying the very careful tracing of the endemic, a few samples in endemic outbreak of the tuberculosis in New Zealand. So I don't know why this is gone, but anyways, so in Bayesian, we don't get just one tree, we get distribution of the tree. So that's why I'm plotting here the summary tree, which is something called the posterior maximum credibility tree, MCC tree. So, and then it's colored by the posterior mean of the proportion of each edge spent on dormancy. And then this bar is the posterior node height distribution. Okay, so in the paper we're basing on, they estimated the active TB is mutating about 1.13 times faster than the latent TB, and then our posterior mean estimated 1.12. Obviously, if it didn't work, I wouldn't be showing it, so you will see that this works very well in this example. And then the expected latency period empirically is 1.27 years, and our posterior mean is 1.3 years. And then the S to E transition rate, if we fit the compartment model, then there's some range in empirical papers, but then our posterior mean is like within that range. And then the paper also reported some putative epidemiological link, and our MCC tree also recovers that putative epidemiological link. Okay, and then the more detail, also we implemented this as an open source package in this. So if you want to take a look, it just got published in PNAS if you want to see more detail. And then current work, there's a lot of exciting extension for this. Obviously, one of the obvious ones is incorporating recombination, but then also in coalescent framework, one of the advantage of using coalescent over birth sampling model is that it can, it's not showing the credible interval, but it can non-parametrically infer the effective population size trajectory over time. So because, for example, in Kim and coalescent model, the coalescent times are sufficient statistics to infer the effective population trajectory. Okay, and then there we have something called preferential sampling. So in epidemiology, so when there's a lot of activity, then the sampling intensity is very high, and then when there's not many activity, then there's not. So that the sampling, we can actually model the sampling process is dependent on the underlying process, which actually reduce bias and accuracy. And then which is most interesting to me is that right now, if you may have noticed, its lineages are transitioning to active to dormant independently, but then if there's some environmental pressure, that you can actually impose some Poisson measure across the lineages so that they can stochastically transition together at the same time. And then like a dormancy is very interesting topic for me. So Gilly said it's a good, important ecological trait. So we also model the gene drive dynamics in plant. It affects on the gene drive spread time, introduction frequency or frequency dependent drive. And then the also spread potential that has the implication for biosafety and containment. So I'm happy to talk about any dormancy related topics as well as development dynamics. And thanks for our collaborator, Andy, Marty, and Lorenzo, and then the lab. If you know us, like we love food, and then the funding sources. Thank you.
All right, and can the next speaker come up while we ask a question?
Yes.
Yes, I have a question about applications. So can you just throw this, I mean, there isn't that much plant data perhaps, but on bacteria, can you just throw this technique on every species of bacteria for which you have enough data and compare how much dormancy they all do?
Yes. So as you may have.
We're not on the microphone anymore.
Oh, sorry, I just took it off for other people.
Yeah, just hold it. Yeah.
John, come up.
Oh, yeah. So
You hang on the microphone while he sets up.
Okay.
There we go.
I didn't model recombinations. A lot of plant species has recombinations, so that's what we're working on right now. But then also plant species has their dormancy is like very short compared to the bacteria. So for bacteria, as long as they're clonal, technically yes. And then also their transition is not correlated, like I said. So if they're correlated under environmental pressure across lineages, if there's some global environmental shift and everyone goes to dormancy at the same time, then we have to use the current model that we are working on. But other than that, and also there's some high frequency spectrum summary statistics based method to detect or just to test the presence of dormancy from the genetic data. So this is more, I guess, like overkill if you just want to detect the dormancy. This is just infer the parameters as well.
Yeah, you just detect and get a dormancy parameter and go across the trees of bacteria and say who has how much dormancy.
Yes,
like primitive, you could do that.
Yes.
Great, thank you.